do it again. Yeah, this will be fine. You will get a folder. Get a folder and all the video that. So all right, let's start. So today will be our last lecture about the RNA-Seq data and RNA-Seq data analysis. So basically today I will summarize the key points and the key knowledge points and take-home messages we have in the past 2 lectures. And also we will introduce some advanced topics in RNA-Seq data analysis. And also we will do the hands-on part later. And then I will do, if we have enough time, I will do some introduction about single-cell RNA-Seq data study. So this part is very important. So you see the take-home message. So you can just, I think if you just think about it, and you can link it with future exam. So basically what we have talked about in the lecture 1 is we talked about the RNA-Seq, basic idea of the RNA-Seq experiment, the experiment process and parameters, published the project of gene expression landscapes of cells, tissues, and disease. And also some difference between RNA-Seq versus microarray. And also we talked about the alignment algorithms and including map to genome strategy, spliced aligners, including the 2 kinds of methods. One is exon first methods with an example of top hat, and a seed extended method with an example like star. And then we talked about gene expression level calling and the definition of RPKM, CPM, and TPM. And today I'm also introduce something new. Is named exon union or exon intersection method in assessing the gene expression level for each gene, with considering possible alternative splicing isoforms. And then in the first lecture we have also talked about the DE-Seq. And today I may use the hands-on part to introduce some details about DE-Seq. So no worries about this part now. So I will introduce some details again later. So if you think, okay, some terms I don't know what's the meaning of it, please be concentrated in the next 20 minutes. I will introduce the key points of those methods again. And those are maybe related to the exam, 2 questions. And from the second lecture, in the second lecture, we first talked about the statistical power. But statistical power at least is as in, and also here, we talked about some other methods of differential expressions. If those are in gray colored characters. So I will not raise any questions for the final exam about this part. But I think these are good for you to learn about this. So, and also in the second lecture, we have majorly discussed about the alternative splicing, the types and regulation, and the array-based approaches, and also alternative splicing isoform calling methods, including two major categories. One is reference genome depending one, and also the de novo assembly-based methods. And also we talked about the alternative splicing event inference methods with an example of MISO. So let's just do a review of the major key computational parts or most challenging parts, and we have introduced in the past. So first is about the RNA-Seq alignments. So 3 major challenges of RNA-Seq analysis. First is the short arrays. Second is error rates are considerable because there are maybe certain errors, and also many rates span exon junctions. So this is a key point. So and why we need spliced aligners. And there are some early studies using map to the genome strategy and just with achieve 2 goals. First, annotation, identify of null transcripts from that location of regions covered, and quantification just to estimate the abundance of transcripts from their depth of coverage. Actually, these are the 2 major goals of all the RNA-Seq expression studies. Just the first get the annotation and map the transcripts, call the gene expressions for each transcripts, just annotation and quantification. And what's the first strategy is unspliced. The first strategy we have talked about is unspliced array aligners. It's just a direct map to the genome, and this method doesn't allow large gaps. But for example, like bow tie is used in the top hat pipeline. We will see it later, and but these methods are ideal for mapping rays against the reference cDNA database of quantification for quantification purpose. Because in cDNA you have already known all the transcripts, so just to do the same thing like linear genome alignment. but it's not good for identifying novel transcripts. There's no way to study like the alternate splicing. And and then we talked about spliced aligners. Basically, there are two major categories. First is exon first. So we just for each of the reads, after getting the raw race, we first map the race to the genome, and using the for example, like the top hat, we use bow tie, and map the race to the genome for the race mapped to the genome. We get which exons are expressed there, and then we analyze the unmappable race, and to study the alternate splicing event. It's alternate splicing, possible alternate splicing isoform. And second one is about seed extender approach. So first we just split the read into certain k-more seeds, and then we use the seeds to we use those seeds to compute the most possible exons or genes or genes that the race are from. Each radar is from, and also we can and for the race just map to one exon is okay, and for some race with part mapped to one exon, we may extend it to the, and with the rest of the k-more mapped to the other exon, and we do the transcript stitching and get the whole, get the final alternate splicing isoform call. So, and here is just a summary from a review paper about the unspliced aligners and spliced aligners. And this is an approach of top hat. So good to learn this, good to keep it in mind. So the first step, just map race to the whole genome with bow tie, and assemble the consensus of covered regions and generate possible splice between neighboring exons, and also talk about, and then we, on the other side, we get the collect initially unmappable race, and then get them together to get the final splicing of isoforms. And this is another figure about the approaches. And the second one is, as I mentioned, about the seed extender approach. We split the read into k-mores, and the k-more can help us to seeding or anchoring each of the parts, a certain part of the race, maybe the whole race to an exon, and also the rest part, if it's the read is over a splicing junction, it can help us to anchor the second part of the race to the other exon. And then we have I have one slide about the star pipeline in the first lecture, but that one will not be included in the final exam. So you just need to get the key idea about this, and also keep in mind. So through these lectures, we have talked about a lot of computational issues, how to play with the race, and how to answer specific questions. I hope you can get the major point about this computation strategies, what we can use for different purposes, what things we do. Like sometimes we do graph-based models, sometimes we just do mapping, do alignment, and because in the future, once you have some certain specific questions, if the method, if you're a computational background, if this method, none of the existing methods work, so you can develop yourself, something by yourself. Yeah. So this is about star aligner. Some details is like I think the most important thing you need to keep in mind is about the characteristics. Don't rely, it doesn't rely on gene structure definition, and can be used to identify novel splicing junctions, gene fusion, and poly tail. And it's extremely fast, but require large memory. And also always keep in mind, there are published results, always published results by using simulated data or just the real data to test the efficiency and just how each of the alignment algorithm works well or not. So, but it's the same for all the other methods I have introduced about. So, because if this topic is quite mature, so if there are like more than 5 mature methods published available in the public domain, so you can always find some paper about the testing or just assessment or just a survey of the effectiveness of the methods. And then we talked about the gene expression quantification. So because once we get the counts aligned to the genome, and we can call the counts mapped to the number of counts mapped to each of the gene. But these counts are still not the relative gene expression abundance, because through different samples, the counts are biased by the total number of by the total number of counts in each experiment, each sample. And among different genes, it's biased by the length of genes. So we have introduced the idea of RPCAM and also the same one with FPCAM for parallel data. So keep in mind, just please remember this formula. So this is, but the 10 to the power of 9 may not be that important, but no, the C is on the upper side, and the C is the total counts mapped to one gene, and N is the C is the count, number of counts, the counts mapped to one gene, N is the total counts in that sample, and L is the gene mass. With this normalization, we can get the relative abundance, and by this definition of each gene in each sample. And also we have the definition of CPM and TPM, please keep in mind. And also CPM, we don't normalize it to gene length. But sometimes when we just need to compare the differentially expressed genes among different conditions, between different conditions. So we don't need the gene length. We just do for each gene, we do the sample to sample comparison. So we don't need the gene length here. So, and here is something new. So I think I mentioned, because there will definitely be a reason that I introduced this part here today, and which is we have about the things about the sub-knowledge point we have passed. So here is this one is very important. It's you need to really keep this in mind for your exam. So it's about gene expression adjusted with isoforms. So once we get the counts mapped to the gene, so and we know the genes is not just a simple line with some exons. And we know there are some certain isoforms. And we know that, and sometimes we know what each isoform is. So there will be some cases, so how to count the gene expression? So we just want to count the average gene expression. But for some exons, for some isoforms that with relatively small amount of exon, for example, just one exon, but this isoform is with huge gene expression. But for some other isoforms that with very long, this is very long, and for this isoform, the expression level of this isoform is relatively low. This is the first case. And when you take the RPCAM, you will use the total counts. If you use the total length of the gene, you will use the counts. The major number of counts are from the first isoform, from the short exon. But you divide this value by the total length of the gene. But for the second case, if the gene expression of the second isoform is very high, it's the same level as the first case in the first sample of the first exon in the first sample. But here you divide it also by the total length of the gene. So then you will get bias. So because the number of counts can be mapped to the number of counts from the shorter exon will definitely be smaller comparing to the total number of counts get from the longer exon, the longer isoform. So we will see one example later. But here we have two methods two kind of rules to adjust the gene expression with no isoform structure. So that is exon union method. The second one is exon intersection method. So exon union method means if we know there are two alternate splicing isoforms, we count all the counts together and divide it by the total gene length. And the second one is exon intersection method. We just use the race mapped to the exons that are intersected by that are contained by all of the isoforms, and then normalized by the length of these exons. So this example will be more clear. So here are two conditions, condition A and condition B. And this is the ground truth. We actually, from the experiment, keep in mind, we cannot observe, we cannot say which race is from which exon, we don't know. But this is the truth that happens here is for the isoform A, the length of isoform A is L. And the length of isoform B is 2L. It's just 2 times as the length of isoform A. And this is the structure this is the whole gene structure. And this is isoform A that looks like, isoform B looks like. And this is the exon union model. We use all of these exons. And this is the exon intersection model. So we just use the exon, the intersect part. So if in condition A, this is the case. We have 6 rays from the isoform A and 4 rays from isoform B. So So and in condition B, we have in total 10 rays from isoform B. So if we use the union count, so this is 10 rays from from B, 10 rays from 10 rays on gene A, and they divided by the total length. So the fold change will be 0. Log fold change will be 0. But if we just use the intersect counts, so intersect counts we have here and here. So the number of rays mapped to the intersect region. Here is 7, here is 8. So log fold change is 8 divided by 7. But the true log fold change, how to compute it, is 10 divided by L total, and just normalize to this value. 6 divided by L is the number of rays from A, and 4 divided by 2L from the number of rays are normalized by the length from condition from isoform B. And in this one, and in B, the case is that all of the counts are from the isoform, the second isoform, and isoform B divided by the length of isoform B. So here you can just ignore here. This one you just put it aside a little bit, but you just first try to understand this part, how to compute this part. So for the second case, just another extreme case. So in condition A, all of the rays are from the isoform B. And in condition A, in condition A, all the rays are from isoform B. In condition B, all the rays are from isoform A. So if you use union count, this is the total number of rays. Here are 1, 2, 3, 4, 5, 6, 7, 8 rays. And here are 6 rays. It's 6 by 8, which means this one is downregulated in this condition. The gene is downregulated in this condition. But if we just use the intersect count, just the intersect part, this part, so the number of rays mapped to this region is 5 to 5. So like I mentioned in the first time, all models are wrong, but some are useful. We can see none of these values are exactly the same as this one. And even this one is the extreme case. The true fold change actually here is this gene is upregulated in B versus in condition B versus A. But here we count is downregulation. It's by the union method, exon union method. So please keep this definition in mind. So, and this is definitely what we call the gene expression. So sometimes there are default settings. You will ignore this. But actually for RNA-Seq and for some other methods, I know there are this adjustment, this assumption, you can change the assumption. And here's a clear definition. Exon union method use all of the exons and underestimate expression level for transplanted genes. And exon intersect method is use only constitutive exons and reduce the power of differential expression. So, and if you feel this part is not that clear, so please go back and really focus on this part and check about the definition. Yeah. And then we talked about the RNA-Seq differential analysis, and we talked about some methods different to DE-Seq, just some methods from microarray data analysis. What we have done for the microarray data, and we can continuously use this in RNA-Seq data study. But like I mentioned, if the number of samples is really low in each condition, so some of the methods with relatively low statistical power, and some of the methods with relatively wrong assumption of the distribution. And then we focus on the DE-Seq. DE-Seq is very important. We talked about this in all of the 3 lectures, and today the hands-on part will be majorly focused on this one. So we have talked in the last lecture about how to increase statistical power. One way is to get a rational data distribution assumption. So to derive the distribution there, we start from binomial distribution with the assumption that N is the total number of rays we observe in a sample, and P is the probability sampling of rays from a gene. And then with the P, N is very large, P is very small, and we approximate the distribution to a Poisson distribution with one parameter, but then we found some dispersion issue. That means when we have the real data analysis, I will show you the hands-on part. So the variance of the counts will be always larger than the mean of the counts. That means the Poisson distribution is not, the Poisson assumption is not that correct. So this is with a larger variance comparing to the mean. So then we have introduced the negative binomial distribution definition, and just add one parameters that can make the distribution be flexible enough to enable higher variance compared to mean. And here, but we get a rational derivation or kind of our explanation is that we assume the lambda follows a gamma distribution. It's a variable follows gamma distribution through all the samples. And with this assumption, and with the counts of each gene follows a Poisson distribution, and the final distribution of the counts for each gene and each sample will follow a negative binomial distribution. It's kind of if we put gamma distribution as a prior of lambda. If you know something about this terms in statistics, you can regard that we give a prior gamma distribution for lambda, and the final posterior distribution of the counts will follow a negative binomial distribution. And here, this case for who has learned about statistics, about the law of total variance, so you may know this formula. So that if y, which is the lambda in this one, but definitely you say this is gray, so it's not included. This part will not be included in the final exam. So, but here I just want to let you know that if y is just a variation of the, just for example, if we still assume the Poisson distribution is from Poisson lambda, and the lambda, if lambda follows a gamma distribution, so that means the variance of the counts equals to lambda, and lambda depends on x, which is this prime. It's just shows its prior distribution as a gamma distribution. But here, the key point I want to think about is that if lambda is a fixed value for Poisson distribution, Poisson lambda, the variance will be lambda. And if lambda can be varied a little bit, but not fixed value, just a little bit with a like big peak, very sharp peak, so the variance will be larger. The final distribution of the Poisson, and if you take random lambda from this distribution, and then take the Poisson, and then use the random variable lambda to randomly sample the lambda to generate the Poisson distribution, and you draw random numbers from this Poisson distribution, and if you do it multiple times, the final distribution will with a larger variance compared to a fixed lambda. Just think about, if you don't catch the point, just think about an extreme case. So still Poisson distribution of lambda, and lambda can be varied just with a huge variance. Lambda can be 1, lambda can be 100, lambda can be 1000. And if you draw a random number from this huge distribution, you will get a big variance of lambda. And then if you, for each of the lambda, you get a Poisson distribution, you draw one value, one random number from this Poisson distribution. The final result will be, if you put all of the randomly generated final Poisson random numbers, the variance will be huge, because the lambda is with a huge variance. So the key point here I want to, I hope you can get is that with enabling lambda can be varied, and with lambda with a variance, the final variance of the Poisson distribution of our random, the read counts, the final variance will be increased. And as shown in this figure, so also this formula. So if you do some computation, this value will be equal to lambda, and this value will be a positive value. So then you will get some value larger than the lambda. So that's how you using this two-step, this hierarchical model to increase the variance of the lambda. So, but this part will not be included in the exam, but I hope you can get this point. And the second lecture, we have talked about the alternative splicing. So we talked about the different types of alternative splicing, and we have talked about the regulation of alternative splicing. And please keep in mind, keep this biological mechanism in mind. And also we have talked about like array-based methods to study alternative splicing. And like this splicing junction array, this defines certain probes. We can study some we can quantify different alternative splicing events. And also and we can, but the array-based method really demand we have prior knowledge about the isoforms, about what the isoforms looks like. But there's no way to detect, to identify, the power to identify novel alternate splicing isoforms will be very low by using array-based method. And then we have talked about the computation strategies for alternative splicing study of RNA by using RNA-Seq data. So we have first talked about the isoform-based approach, which means reconstruction of the transcriptome, reconstruction of all the isoforms. We have talked about genome-guided reconstruction method with an example like cufflink, and also genome-independent reconstruction method like Trinity and Bridger. And also we have talked about the alternative splicing event-based approach like MESO, just other than compute to fully reconstruct the isoform, the transcriptome, we just estimate the event or the probability of the inclusion, exon inclusion for each alternative splicing event or for each exon. So just do a review of the cufflink with 5 steps. First one is alignment by using top hat, for example, by using top hat, by using splice alignment. And the second part is to identify the rays that are not compatible to each other. The definition of compatible is that these rays are definitely not from the same isoform. For example, because we have mapped it to the genome, we know, okay, this one is with, for the example of this two, this one is with a mapping region here. It's like mapped to, because just imagine there's a line of genome. So this race is mapped to the genome with a region mapped to here. But this one is missing that part. So that means this two definitely have an alternate splicing happens here, exon inclusion happens here. So, and then with guys the rays constructed whether each any two rays are compatible to each other, the cufflink further construct an overlap graph, and the overlap graph is constructed based on one criteria. First is each node on several criteria. First, each node is one ray, and each edge just links the overlapped rays, and the rays are overlapped and are compatible to each other. And then we make identify from and this one, because we know the direction of the gene, we know so then we get the overlap graph will be a acyclic graph. And we know, and we can identify the mutually incompatible fragments on this graph. And there is a graph theory, there's a theory in graph theory that the number of the incompatible fragments here is exactly equal to the number of paths we need to cover the whole graph. The path is just one directed path. And then the cufflink will enforce the minimal number of passes needed to cover the full graph, the full overlap graph. And then each of the paths is just corresponds to one possible alternative splicing isoform here. And also please keep in mind that this graph is can exactly reflect the structure of the possible alternative splicing. So probably just possibly in the exam, I may get a question that with observed, this is an observed overlap graph, and what is the most possible alternative splicing pattern? And then it estimates the transcriptional abundance by using likelihood function, just by using the input of the grid counts and also the constructed graph. And also we have talked, and here is some characteristics of cufflinks. It requires spliced alignment, such as top hats, and capable for identifying and quantifying isoforms from RNA-Seq data, and can be used on both single end and paired end data. And and there's following algorithm for differential expression test is available named CuffDiff, and it's also open software, open source software. And also we have also talked about genome independent reconstruction. So the challenge with genome guided reconstruction is that it depends on the current way to reference alignment. And if we will talk about gene fusion later, but in cancer, so cancer genome is highly varied, may not be, or the cancer chromosome may even be highly varied compared to a normal cell's chromosome. So the reference genome may not work well for cancer. So sometimes we may need just a genome independent reconstruction for cancer study. So the genome independent reconstruction method, first is use rays to directly build consensus transcripts. No alignment is required. It's called assembly. So and it's great for species without a reference genome. For example, just regard cancer as a new species. yeah, the cancer genome is still quite similar to normal human genome. Yeah. So and also there are multiple tools we have introduced about Trinity and Bridger. So Trinity is a transcriptome assembly method with 3 major steps. So first, the inchworm one is highly memory consumable, and second one is cry cells and butterflies. So we will talk about details in this one. So first, for each of the rays, it's still split it into k-more, and using the k-more to link to the deep-brain graph. So basically, they get the k-more with the highest count, and then they get a k-more that can be linked with this k-more, like shown in this figure, with just one position switch, just one position slide from the first k-more, and get slide from one position, and among all the k-mores, they select the k-more with the highest counts, highest number in the data, and then they do this by using this way. They construct the deep brain graph, and they can do it for if they didn't meet the final, just if they didn't clear all the rays, all the k-mores, so they can continue construct one deep brain graph, second one, third one. Each of them will form one contig, and then they may use the second process using to overlap linear sequences by overlap of the k-1 mores to build the graph components. So basically, just imagine, so none of the two sequence RNA contigs can have a k-length overlap. The reason is once we construct it, all the k-mores are used. The k-more are eliminated from the pool. So, but they can have the longest one is k-1 more, k-1 more overlap. And if there's really truly alternate splicing happens, that means the branch should exactly match to just one original contig and the k-more with a different alphabet in the final spot. That is the k-1 more overlap compared to the original contig. So and by using this, you can build this graph. After you get this graph, you can call it kind of shows, this one just reflects the real. We can't directly get it, but this one just reflects the real splicing isoforms. Like this one is the light blue one is one, dark blue one is one, red one is one, and also the pink one is one. And after you get this figure, we do butterfly step. You can just, this part can be really, it's kind of adjustable. Like we can just call like all the possible paths from the beginning to the end, from the top to the bottom to get all possible alternate splicing. And in Trinity, please keep in mind, the gene expression level is not used there. So nothing about gene expression level based adjustment, just the is. Just if there is one path, just one isoform. And there's some details at least there, so good for it, I think. And also please keep in mind, there are always some methods to test the performance of different alignment method. And also, I think I missed one slide here. So let me see. Oh, probably I missed the Bridger one, the Bridger slides, but Bridger will not be included in the exam. But I just want you to know, want to keep in mind that the Bridger is a method to bridge the Trinity and the cuff link. So basically, once they get this figure, they compute the minimal number of passes they need to overlap this figure. So, and also, and here is also another review about the two different methods, one's genome guided method, one's genome independent approach. And oh yeah, here's Bridger, sorry. And the Trinity is very sensitive and with high force power, and they didn't use like the gene expression level like I mentioned. But Bridger is just a kind of improvement of the Trinity. But Bridger also uses a strong assumption, like it be very specific, like cuff link, and to find the smallest number of paths, the minimal number of paths that can overlap the graph, the overlap graph. And then we talked about a kind of method named the alternative splicing event identification methods which treats the alternative splicing from another angle. So sometimes we don't need to call all the isoforms, especially if we just want to study what regulates alternative splicing. And also sometimes is we just want to see the difference between two different, we just want to use, say like supervised training or unsupervised training. We don't need to know what exactly isoforms is. We just need to get the data to some possible biologically rational events. We just do it. We can just use this kind of methods. It's, because the model is relatively simpler, so the force discovery will be lower, I think. So here's the idea: is that we get two isoforms. We just want to compute the percentile, the percentage of the exon inclusion of this one. So we have introduced a method like meso, and the method is like give a define of percentage spliced in. Is this side. So and it denotes the fraction of mRNA that represents the inclusion isoform. That's this one. So and meso, here are some characteristics of meso. So it uses inclusion rates, exclusion rates, and also constitutive rates. So sometimes you may think, okay, constitutive rates is not that important, but actually if the alternative splicing event is more complicated, the constitutive rates can be informative to, you can just write a maximal likelihood function by including the constitutive rates, by thinking about that the inclusion and for this case, just the inclusion and the inclusion and the exclusion rates, the total of this rates should be relatively equal to the depth or density here, and also same to here. So the constitutive rates are not non-informative. So the method actually creatively assigns the constitutive rates back into different events using Bayesian method. So also in method we can test the difference of splicing pattern by using a Bayesian-based Bayesian factor. Actually, it's also ratio just. And okay, so here is just a review of all the topics we have introduced in the past 2 lectures and all the major computational parts. So any questions here? So okay, so this part I will post this part on Canvas. And for some of the key issues, I know there's I didn't record the first lecture, and the first half of the second lecture, when I uploaded it to YouTube, it's kind of some error there. So I may redo some of the lectures, video record of the lectures, and upload the key parts. It will be shortened than the original one, but I will upload I will redo the most essential parts and upload it to the Canvas or to YouTube. Yeah. So let's talk about some advanced issue in RNA-seq data study. So one thing, the first topic I want to talk about is gene fusion. So a fused gene is a hybrid gene by drawing parts from 2 previously separated genes at transcript or genomic level. So just by the most straightforward definition, just 2 genes fused together. So this figure shows the genome in cancer. So the original genome maybe looks like that. But with the progression of cancer, the genome, due to just possibly some oxidative stress, something that may induce mutation in cancer environment, the cancer genome will be much unstable. So unstable genome is one hallmark of cancer, and the genome will look like that. And some of the chromosomes may be just translocated to each other, and just some genes will be fused through this process. And one famous example is about the current BCR ABL1 fusion gene in CML, chronic myeloid leukemia, myelogenous leukemia or myeloid leukemia. So CML is one very famous case, and there's a drug named Gleevec just got directly to treat this disease, and it happens in more than 95% of the CML. And now the CML, with identification of this event and with the development of the drug, so now CML's treatment effect is very good. So, and also there are some other reported gene fusion in other cancer, but the prevalence is not that high. So, And there are several mechanisms that cause the gene fusion, like the inversion and also translocation and large division insertions. So you can just wiki it or put a small figure here. You cannot see it clearly, but once you get the slides, you can enlarge it. So about the mechanisms, and also sometimes the gene fusion is gene fusion may definitely have impact to the cell, but sometimes just imagine if the two genes are not used in the cell, and just fused, and once it get fused, for most of the cases, the protein cannot fold, and the protein just degraded. So it will just lose something, but sometimes it can be very bad. First, the genes, the promoter and the enhancer elements of one gene are aberrantly juxtaposed to a proto-oncogene. This is just a definition, some case about the proto-oncogene to an oncogene. So if something happened in the promoter region, so the oncogenes, for example, MYC, will cause alter the expression of large overexpression boost of those oncogenes and trigger the downstream cell cycle, cell proliferation signals. And also, the two fused genes may cause may get a new function. For example, like certain activation of certain tyrosine kinase or certain other stuff. So to study the gene fusion, and one ideal method is just definitely NGS, the next generation sequencing, and especially the RNA-Seq. So we can use the paired end race to study the gene fusion. For example, we just get a paired end race, and the two fragments mapped to different genes. That means the gene, and with a high frequency observation of this one, we can infer, okay, there will be a gene fusion there of these two genes. And it's good to increase the ability to detect fusion events compared to single end. But the problem is that we don't know where the fusion. If we just use this kind of paired end sequence without a fragment locate on the fusion point, on the break point, we cannot know where is the true break point. And using single-cell arrays is provide rate evidence of the fusion events in one base pair resolution, and the longer race and can just get where is the breakpoint, but definitely longer race will be certainly help here. So regular alignment will definitely miss the fusion events because we just assume the reference genome is correct. But large gaps on a different chromosome cannot be identified by regular alignment. But there are some other analysis strategies. First, definitely de novo assembly can help. We can just do de novo assembly to cover some possible gene fusions. And after we get the assembled gene fusion cases, some cases, the assembled cases, we can just map it, just still check if this is a gene fusion or not for each of the mapped contig. So aligned assembled contig. And also paradigm based up, there are some paradigms based approach based on statistics and like diffuse, and also some improved alignment based approach like top hat fusion and Java. Java is relatively new one. Just added this because most of the parts are I just used Dr. Liu's past slides, but this one I just found this one just published recently, and it's with a top performance. I may want to introduce a little bit about this. Yeah. So basically, the paradigm race-based method is quite straightforward. You just get the high frequency, some race just spanning two genes with high frequency. And we can define certain threshold for that, and a statistical method to test about that. So this is the diffuse. And the step one is just look for discordant alignments using spanning race. And second is target search for split race using dynamic programming to find the fusion point. So we get enough frequency, we get the number of rays that span the two genes high enough, and then we check about the rays that exactly covers the possible breakpoint. And also another single and base method named the top hat fuse is just the similar idea that for array is split into three parts. For 75 base pair arrays split to 25 base pair arrays, and then it will just map the different parts to different. If we see a first part and third part can be mapped to different genes, we may infer, okay, this may be correspond to a possible gene fusion, and we check the middle part. If the middle part can be mapped to a fusion point, and this one you can see this. how to split it into 3 parts. The criteria to split it into 3 different parts may highly affect the effectiveness of this method, the performance of this method. And then I want to talk about Java. So this method is recently developed, and I found it. I just did some literature search about the performance testing among all this kind of gene fusion analysis methods, and I found that this method really performs well compared to other methods. They still just do mapping and for the mapping and to assemble identify and also identify the rays that are kind of aligned to multiple regions, and count the rays that support the break, and also do some just do the similar approach as alignment and the breakpoint identification. But I think the most important here is that they get a final step to scoring each of the gene fusion events they have identified, with by using some certain criteria like evidence of rearrangement from public domain, like number of breakpoints aligned to exon boundaries, and also like how many spanning rays, how many spanning pairs. So how many pairs? This is spanning pair. So just the 2 fragments mapped to two genes. And spanning rays means just 1 fragment that's mapped to the breakpoint. And then we use these values to train class to just give the scoring or the confidence level for the called gene fusion. And here just a summary. We have this kind of methods. And there is definitely, this paper just compares the different fusion transcription detection methods by using RNA-Seq data. So you can see the Java with a high sensitivity here. And but they have also some downstream testing. So I found that Java is highly time consuming. So if in the future, if you need to study about the gene fusion, you need to just read some of this paper and evaluate which method you may need. And the second topic I may want to introduce about here is allele-specific expression. So basically, allele-specific expression, just by the simple definition, just for one gene, just one allele with different expression level compared to the other one, and it can be seen by if these genes carry a certain non-synonymous SNP over in its exon, in its coding region. And also, it can be caused by different reasons. Like, first, definitely something happened on the gene body. The trans part is definitely can cause different expression. Like they may have different methylation level, and also something happened on the promoter region. The difference of the 2 alleles may just result in the different allele-specific expression. And allele-specific expression this event may result in different things. This kind of the different concentrations of expressed proteins, different concentrations of cytoplasmic mRNA, and it's highly tissue specific. We'll talk about this later. So, and also sometimes it can be a good thing or can be a disease related thing. But we will see the difference of the allele-specific expression in most cases are not that significant or high. So, and I'm not studying this part, so I can just do brief introduction about this. So, and we know the gene expression is a complex trait that is influenced by the cis and trans-acting genetic and epigenetic variants. So cis here is cis, just a promoter region, trans on the gene body region, on the coding region, and can be the genomic variation and the methylation variation. Or any variations there will have possibility to cause the allele-specific expression. And if we really want to understand if there is a significant allele-specific expression or difference or allele different expression, we may need to integrate genomic and transcriptomic data to understand it, because we need to know which SNPs are there. So, and also some biological hypothesis is that, so for the different tissues and different under different conditions, if we have some possible allele-specific expression, this mechanism for some genes is kind of increase the flexibility of our cell, of certain cells, certain tissue. And with the environment changes, it can be responsive to that. And here is some public, old study. It has identified 20 of the cells shows 1.4 difference between alleles, and 30 of the cells shows 1.2 difference for change. And so that means allele-specific expression is generally existing there, and but the phenotypic effect of allele-specific expression may not be obvious. So, and this one is like it's kind of the dependence on expression are really too small. So it cannot change that much, because the gene expression will then get into translation, and the allele-specific expression is not can hardly form just a strong driver for significant change. And also, allele-specific expression is highly tissue-specific. Recently, there is a Nature paper about the review of the allele-specific expression in mammals, a lot of mammal species, mammal cells, and they found first very tissue-specific. This is the most important thing, and can reflect some environmental effects, but it really need to. It's not a very fixed or very well studied field, I think. Yes. But there are some technology to study allele-specific expression. Definitely, we need to use the SNP. The SNP is on the gene body and on the introns to see if we have the DNA sequence. We have the DNA sequencing or the genome information of the difference of the two allele. So we can definitely use this information to infer the allele-specific expression. And also some other, this technique I just put it here. It's good to read, I think, but I may skip it due to time issues. Padlock-Seq assay. So with certain design, can study the allele-specific expression. And also the most straightforward idea intuition here is that, so we can use RNA-Seq experiment and integrate with the DNA-Seq with the genome information to test use simple binomial statistical test to test if the risk from one allele is significantly higher or lower compared to the risk from the other allele. So, but they may need the hundreds of risks per base pair, or especially map to the base pair with the SNP, and to enable enough statistical power. Okay, and then we talk about RNA editing. So RNA editing is a molecular process through which some cells can make discrete changes to specific nucleotide sequence within an RNA molecule after it has been generated by RNA polymerase. So I don't know if you have heard the news about recently the CRISPR-Cas13 can do RNA editing. So it's recently published by Dr. Feng Zhang's group from Broad Institute. So but this one here, the RNA editing, the definition is not about that. It's not about the CRISPR-based editing. It's just about some enzymes encoded in human that can do the RNA editing. So there are some, it's kind of after the transcription, and after the RNA editing happens after the transcription and before the translation. So, and with on the mRNAs, with inserted enzymes may alter the one certain base of one microRNA of one RNA to another base, just by the simple definition. And it can be synonymous or non-synonymous editing and cause final same protein or different protein. And there are two typical RNA editing. One is A2I or A2G editing. The second is C2U editing or C2G editing. So the adenosine will be changed to the inosine, I think, inosine, and the inosine will behave like a G guanine in the translation. And here are the two enzymes that catalyze the RNA editing process. And here's just a possibility, but how frequent it? So people have done some attempt to see the overall trend of RNA editing. It's like the allele-specific expression. There are also works about the prevalence of RNA editing in different tissues or species, something like that. Yeah. And also the best power you get when you directly compare DNA sequence with RNA sequence, and it requires really deep and it requires, so once you have the deep sequencing and the RNA-seq, the steps you don't need that much, or you will, if you don't have the DNA sequence, so you really need a large deep sequencing. So there are some published study about the RNA-seq, RNA editing, and we can see at least the result is at least 10% of risk covering a set differ from RNA, and around 10,000 exonic sites, and with this one, with RNA editing, and 4,000 known genes with this RNA editing and with this criteria. So it worth to read this paper. Yeah. And here is summary shows that the RNA editing does exist, and the canonical editing is just this two mechanism. Oh, but please keep in mind, so I just excluded one slide from before, but this one is like it's under a big debate, I think. So is that some published results have shown that there are more general RNA editing events, just like not only the canonical tool, but there are more other events. But you may really need careful validation about that, because it can be caused by the false discoveries or the false mappings or something false, or just it can be because you do it in tissue, the genome you have may be biased because you just get a tissue level genome. Genomic sequencing. So if there are a certain proportion of the cells are with a varied genome, but you cannot call it by the DNA sequencing. But this small proportion of the cells are with really extremely high expression of those RNA. You may really get a lot of RNA are with identified as an SNP compared to the genome, and like that. And compared to the mRNA of the rest gene of the rest cell, yeah. So it can be there. Please always keep in mind. So if you use box cell or tissue-based studies, there will be always bias. And the last topic I would like to talk about is non-coding RNA. So actually, so this is from ENCODE project. 70 to 90 of the genome is transcribed into RNA. And non-coding RNA is a functional RNA molecule that is not transmitted into protein. So there are multiple non-coding RNA species. But today we'll just talk about microRNA. So the microRNA, you need to keep in mind three parts of the process of microRNA generation. They have three forms. Pre-microRNA is like with a really long mRNA form, just form this hairpin structure. And the pre-mRNA is just with cut of it's transcribed by the RNA polymerase 2 or 3. And with the enzyme cut here, with some cut here, cleavage here, it may form just a 60 to 100 nucleotide pre-microRNA hairpin structure. And then with further cleavage, after the transport out, export from the nuclear, with further cleavage, it will form the mature microRNA and can regulate the gene expression, can have mRNA target cleavage, can just regulate translation, or just a lot of further biological or biochemical reactions. So, and there are a lot of microRNA based biomarkers for cancer study, for all other disease study. But please keep in mind that once you use microRNA is different to gene expression. Gene is really long. So this is just what we did find in my lab. What we did find in my lab, in my study is that, so once you found some microRNA, so be careful. Because there are existing database like TCGA, like also a lot of published data. So just see the variance of those microRNAs in other data, to see if those microRNA, the variance is really as small as what you have identified in your study. And just to understand, so one thing I have observed about that is the variation among microRNAs through different age groups of patient age groups, different tissues, different cancer subtypes are very high. So it's much higher compared to mRNA. So please be cautious about this, about the stability of microRNA, especially what can be reflected in your data. Yeah. And here is a microRNA detection procedure. So the first step is just get the microRNA. So we just select get from the sample by using a certain just by the size selection. Just get the transcript of a certain length, and then we put adapter on it on both sides and do PCR and do sequencing. And usually it's like 35 base pair arrays because the microRNA is really short. So and with adapter. And here is alignment-based method. It's just we get the sequence, and we remove the adapter, and we align it. We just cluster because microRNA, the total number will be very small. We just we get it into unique sequence clusters. We have a lot of duplicated race or sequence tags, and then we align to the known microRNA database, and to see the gene expression level of microRNA. And also the second way is the second kind of method is to de novo detect the microRNA. It's different to de novo detect alternative splicing, because detect the microRNA is like the alternative splicing is kind of straightforward. Is we just need to identify the key structure in alternative splicing is the splicing junction. But the key point here to identify microRNA is this structure, the hairpin structure, about what the knowledge we have from the precursor, mRNA precursor or pre-mRNA to the mature mRNA. If this hairpin is really forms the precursor of microRNA, and this is a true microRNA, the observed race counts should be having this distribution. A high distribution here about the microRNA, and really low distribution here, because here just degraded about that the race can be mapped to this region or this region. And also, if it's observed result is like this one, so also a hairpin structure, but it's quite uniformly distributed through the hairpin structure. We may think, okay, this will be with relatively lower possibility to be a microRNA. So with this workflow, there is a method and also existing package named MIR-DEEP. And so basically the procedure may just still get the deep sequencing, discard the race that mapped to many possible genomic loci, and use the sequence to access potential microRNA precursors from the genome. Something like that will be removed, and identify the pattern, like with high expression over this area and low expression in the rest area. And also there are some microRNA sequences that have frequent variations, which is named isomeres. That means which is that the mature microRNAs has significant end heterogeneity. So and there's also an isomere database about that for our microRNA study. And and also please keep in mind the process of generating these isomeres may not be random. And you can definitely check more definition about the isomere from Wiki. So that's all about the first part today. So let's take a 5 minutes break. Yeah. So and feel free to ask any questions. Yeah.